Hi everybody. Today we're going to be talking about muscles. So hang tight, we're going to get started on it. All right. Like I said, we're going to be talking about muscles, how they contract. First we're going to talk about skeletal muscles, then we're going to go over and talk about smooth muscles. We're going to talk a little bit about some energy sources that our muscles rely on. We'll talk about how they work. We'll talk about some muscle sensory receptors and things. Anyways, I've already kind of drawn things out just to kind of keep this video as short as possible and as engaging as possible, so that I'm not taking up too much of your guys' time. First thing we're going to talk about, this is kind of a picture of the brain right here. Okay, so from the brain we have these upper motor neurons. They come down and they synapse with these alpha motor neurons in the spinal cord, and these alpha motor neurons go to these what are called motor units. So these are just bundles of muscle fibers that are innervated by an alpha motor neuron, right? So I'm going to kind of take you here. We're going to follow this arrow, right? So we're going to look at the neuromuscular junction, which is this area where the neuron and the muscle kind of get really close together, and there's that synapse right here. Okay, so as we've talked about before with action potentials and all of that stuff, we have some sodium voltage gated channels, so V and A. We also have some potassium gated voltage gated channels here. As these get as they open and depolarize the neuron, it causes an action potential to go down the neuron. Once that neuron gets to this axon terminal, it will activate these voltage gated calcium channels. So calcium will rush in. Okay, and then that activates the snare proteins. So these vesicles, which are right here, all the little blue guys here, they're all carrying acetylcholine. So when calcium comes in, it causes the snare proteins to react, causing exocytosis, right? So then we're going to see all of this acetylcholine dumping out into the synaptic cleft here in the neuromuscular junction. Once they combine here, or they're not going to combine, but they're going to attach their ligands to this ligand-gated channel here on the muscle cell. This is a nicotinic 1 receptor. Okay, so when these acetylcholines bind, you're going to need 2 of them for these N1 receptors. When the 2 of them attach, the gate opens, and then we're going to have a huge influx of sodium because the driving force of sodium is really high, right? But the thing to remember is that when you have an action potential, it only propagates along the membrane, right? So muscle cells have what are called T tubules. I'm not sure if I spelled that right. Tubules. There you go. Anyways, they're kind of like projections into the muscle, like something poked in, right? So it carries this signal down along the membrane here, right? So that so that we can have our calcium coming out into the muscle, right? So what happens is, as the action potential propagates along the membrane, it's going to come here and it's going to reach this voltage gated channel right here. And this is called DHP. I can't remember the huge long name for it, so we're just going to call it DHP in this video. Okay, but it's a voltage gated signal receptor. It's not actually doing anything other than sensing the action potential. Okay, but it is linked to ryanodine, or RYR, and that is this green gate that I've got drawn right here. Okay, so when DHP senses the signal and opens, ryanodine is a mechanically gated channel. Okay, so when DHP opens, it causes the ryanodine channel to open as well. So I'm going to take a little closer look at it down here. Okay, so again, our action potential has propagated, right? It reached DHP, DHP opens, ryanodine opens, and then calcium is going to rush out into the sarcoplasm. Okay, so this place where all the calcium is, I've got it labeled SR, and that stands for the sarcoplasmic reticulum. And again, like I said, that's where all the calcium is stored inside of your cell, or at least the majority of it. Okay, so when this happens, when DHP and RYR open up, calcium spills out and it goes all over your muscle cells. Okay, and so when you think of muscles, you just have to automatically think calcium. Calcium controls muscle contraction. They're always linked together. So that's a really important thing to remember when you're thinking about muscles. Okay, so again, we have these things called T-tubules. They're like little perforations down into the muscle so that our action potential can get close enough to this SR to allow calcium to come out. Because if we didn't have this T-tubule, then our action potential would never reach the SR because our membrane is so far away from it. Okay, so that's what the T-tubule's job is for. That's what its job is, I mean. So now we're going to talk a little bit about when calcium spills out all over the muscle fibers and stuff, okay? So we're going to come kind of over here. We're going to talk about, so these are the muscle fibers that I've got running down through here, okay? So just like this, those are the muscle fibers. They're called the Z-disc. I've got it labeled right here, okay? And then we have two different kinds of filaments running through them. So we have this, what's called nebulin filament, and then we have this myosin filament. Titin. Titin is really big. It's even kind of got a spring. That's why I've got it drawn out like this. That's kind of doing the pushing and pulling of the muscles to contract. Okay, so nebulin is going to be made up of actin. It kind of has, you see how I've drawn on here all these little squiggles? These are like alpha helices running around the nebulin fiber. Okay, and so we're going to talk a little bit about what makes up that alpha helice. Okay, and so it's actin. Okay, so actin is, on the nebulin we have three different parts. Okay, we have G-actin, tropomyosin, and troponin. So G-actin, it looks something like this. Kind of a little circle. It has a spot where ATP is bound. Okay. When we get a whole bunch of them all together like this that I have drawn out, it's known as F-actin, right? But in short, it's actin. Okay. And that's the alpha helices that you see over here. Okay. Now, bound to every one of these actins, we have tropomyosin. It's kind of like a covering. And like I've got written here, it covers 7 G-actins. So 1, 2, 3, 4, 5, 6, 7. I've got this tropomyosin drawn, and it's, like I said, covering 7 of them. Okay, and it's kind of like a barrier to it. The next part of what's bound to our nebulin filament is troponin. Now, troponin is made up of 3 sections bound all together like this. We got troponin T, troponin I, and troponin C. Troponin T binds to tropomyosin. I've kind of shown right there for you. Troponin I has an affinity for G-actin, so it binds there. And then troponin C has 4 calcium binding sites. 2 of them are high affinity, 2 of them are low affinity. Okay, so the high affinity, we're going to hold on to it really well, right? And then the low affinity, they're easier to kick off. They're not always bound, okay? Now it's important to remember, I have it highlighted here, there is 1 troponin for every 1 tropomyosin. So if you think tropomyosin covers 7 G-actins, then that means there's also 1 troponin for every 7 G-actins, okay? So that kind of makes up the nebulin filament, what's going on there. Now we're going to come down to the titin, and it's called it's a myosin 2 filament we're talking about now. Okay, so I've got a really simplistic drawing. I'm not going to go into the more detailed stuff. We're just going to stick with super basic. But myosin has 2 states it can be in. It can be in a cocked state or an uncocked state. If you're familiar with shooting a gun, if your gun is cocked, it's ready to shoot. If it's uncocked, you pull the trigger, nothing's going to happen, right? It has no energy. But when it's cocked, it has lots of potential energy, okay? So we're going to I'm going to show you why or how this all works, okay? So in skeletal muscle, let's pretend that this is our nebulin right here, okay? That line, and then here's our myosin, okay? I'm just going to actually call this line actin, not necessarily nebulin, okay? So we're going to say this is our myosin. We're going to say that this is the actin, okay? Now, myosin wants to be with actin, but like I said above, tropomyosin blocks the actin, the G-actin, okay? So when calcium spills out of the SR, the sarcoplasmic reticulum, and covers the muscle, what it's doing is it's binding to troponin C. Once it binds to troponin C, it moves tropomyosin out of the way, and that allows our myosin head to now come up and attach to our actin filaments. Okay, so when that happens, when tropomyosin has been moved out of the way because calcium bound to troponin and caused a shift, myosin now binds with the actin here, and when we form a bond, we release heat, and we also release enough energy to break this bond with a phosphate. So we lose our phosphate group, okay, it gets kicked off, and then when that phosphate group is kicked off, that causes a conformational shift on this ADP head group. So then ADP gets kicked off, and that causes movement. That's why I've got these little lines here, okay? Now this is known as our power stroke, because that's like pulling the trigger on your gun, right? So you release all of that energy, and it goes bam, it shoots down, okay? So now we've just moved, we just shifted everything, we contracted, okay? So now we're still bound myosin to the actin. Now we're going to add ATP to the system, okay? ATP is going to bind to that head group, and it's going to break the bond. Okay, so then we're going to lose some more heat here, and we're going to create enough energy to break that bond, okay, when ATP binds. Once ATP is bound, we're going to go through some hydrolysis here, okay? So then ATP dissociates into ADP and another phosphate group. Now we are back into our cocked state, right? So it's not too crazy. This is known as our cross-bridge cycle. Okay, this is specific for our skeletal muscle. Okay, so I want to talk about some sources of ATP here that our bodies use, or that we use, I guess I should say. So first of all, we have our cytoplasmic stores. So this is just ATP floating around inside of our cells, right? So there's not a lot of it, so we only get about 1 to 5 seconds of use out of our cytoplasmic stores. So obviously we're going to burn those really quick. Those are meant to just super quick bursts of energy. You know, if we have to contract something really fast, probably something like if you put your hand on a burning stove, just that quick reflex jerk away, get off of it. It's going to use up that ATP. Another source of ATP that our bodies use is creatine phosphate. What it does is it takes ADP, our creatine phosphate comes up, and the ADP steals one of the phosphates off that creatine. Creatine kinase is what takes the phosphate and adds it. So then we get ATP. So it's a little more regenerative, it is regenerative, but it's not limitless. So it is limited. So we get about 10 seconds of use out of our creatine phosphate source. Glycolysis, we're going to get about 60 seconds. So you're going to get about a minute of use out of glycolysis. So quite a bit more than our cytoplasmic stores and our creatine phosphate, right? However, once that 60 seconds is up, we're kind of gassed, right? So all of these are known as anaerobic, the 1, 2, and 3. They're anaerobic sources of ATP. They're just quick, ready use sources of energy, okay? Now, the most efficient, at least the one that can last for a long time, is our aerobic respiration. This can last up to hours, right? So this one's obviously the best, but it's not meant to be used for high energy situations, high intensity situations, I guess I should say. That's probably a better way of saying it. So whenever you're doing something quick, you need to get out of a situation, those fight or flight moments. You're probably going to be using cytoplasmic stores, creatine phosphate, and glycolysis, all of those things. Aerobic respiration, you're using oxygen in your mitochondria to create ATP. This is more like walking or jogging or, things that you need to be able to do for a really long time without gassing yourself out. Okay, so it's not super efficient if you need it in a high intensity moment, but overall, it's going to create the most ATP and keep you going the longest. I hope all that made sense. We're going to move on to part 2. We're going to talk a little bit about smooth muscle now. So smooth muscle is a little bit different. I hope you can see that it looks pretty much the same. I've got kind of 4 of the same drawings here, 4 of the same steps. They look a little bit different, right? But again, this is called the cross bridge cycle. Okay, so, but there's 2 different types of cycles that you'll see. You'll see a phasic and tonic cycle. So we're going to jump in. We're going to talk about phasic first, okay? So with phasic, first thing you need to know about myosin in smooth muscle is that this head group is ugly. That's the best way I can describe it. It's not a nice head group. I just think of it as a lumpy, hard to wrap your head around kind of an idea, okay? But it's not pretty, okay? But when calcium enters the system, I need to mention this. Smooth muscle doesn't have troponin. Okay, that's a big difference. Troponin has the 3 subunits, C, I, and T. That's for skeletal muscle. Smooth muscle does not have troponin. So tropomyosin is still in the way. Okay, but calcium comes in, and it's gonna bind to tropomyosin. And then we've got this enzyme here called MLCK, myosin light chain kinase is what it's. I'm going to write that out. Okay. So what that's going to do is it's going to take an ATP and it's going to steal one of its phosphates. Kinases, they add phosphates to things. Okay, so this MLCK is going to be activated when calcium's in the system. It's going to steal a phosphate off of an ATP, and then it's going to put it right on our myosin group. Okay, and that's going to cause a conformational shift of this ugly head group. So now it's pretty. Okay, so now it's going to be able to come up and bind to our actin filament. All right, so this phosphate, this second one that we added with MLCK, changes the head group, and that allows it to bind to our actin. Okay, and then it's kind of the same steps from there. The binding releases heat, and it has enough energy to kick off our first phosphate, just like we saw in the skeletal muscle. Right, so phosphate kicks off, causes a conformational shift. ATP or ADP then gets kicked off as well. Okay, and again, this is the power stroke. So it causes the movement. Right, you can see that our myosin group is no longer cocked, it is uncocked. Okay, but this phosphate group is still attached. Okay, so this head is still pretty. Okay, so that's important to remember. Now, ATP, we add ATP to the system, and that ATP is going to bind to the head group. And again, we're going to break this bond, release a little heat. Okay, this phosphate's still on here. So head group is still pretty. Okay, so now instead of going back up to here, we're going to kind of bypass this whole step. Okay, so now we're back here. Head group's still pretty. Right? We still have the phosphate, we have all this. So we don't need any of this at this point. Okay? Not unless MLCP, myosin light chain phosphodiesterase, I can't remember how to say it. I'm blanking on that. But MLCP, when it's added in, it cuts off this phosphate. So we're going to lose that phosphate, and we're going to have to start the whole system over. Now we have the ugly head group again. Okay? So we have to go through the whole process of getting MLCK in here, adding that second phosphate. Right? So phasic, where you're going to see that is in places like your blood vessels or your digestive tract, right? It's kind of linked with peristalsis. So just that constant rolling movement, right? Where it's just kind of going and going and going, okay? So I want to show you really quickly what we're talking about with MLCK and MLCP. So MLCK, when it's inactive, it's kind of like a snake biting its own tail, okay? But we have this enzyme called calmodulin, and it has 4 calcium binding sites. So when calcium comes into the system, it binds to calmodulin. Once it's bound 4 different calciums, then it'll come to MLCK, take its tail out of its mouth, for lack of a better way of describing it, and then this snake will now take the phosphate off of ATP and then throw it right here onto our myosin group, allowing it to have that head conformation change, so it's now pretty and it'll make that bond. Okay, so that's how MLCK works. Now MLCP, a little different. When it's inactive, excuse me, rho kinase is kind of chilling in its mouth. Okay, so it's blocked. Right? But when we have a whole bunch of calcium, or sustained amounts of calcium, after a while, that will get rid of our rho kinase, and now we have an active Pac-Man, MLCP, who's going to come, and it's going to chomp that phosphate right here, kick it off, and now we're going to have to go back to step 1 here, instead of just being able to bypass straight to step 2. Okay, so that's kind of how MLCK and MLCP work. I hope that kind of makes sense for you guys. Now we're going to move on to tonic. Tonic is very similar, just with a different placement of something. You'll see it very quickly, right? So same deal here. We have this ugly head group, okay? Calcium needs to come in. Calcium comes in, activates calmodulin. Calmodulin activates MLCK. MLCK takes a phosphate off of ATP and then puts it right on our myosin group, changing our head group to a pretty head group so it'll make that bond. Okay, so then we have the loss of heat. We kick off our phosphate. Kick off the phosphate, causes a conformational change. Kick off our ADP. Now we fire. However, with tonic, what we're doing is MLCP is introduced here very first. Okay, in this same one, we have MLCP. So then we kick off this phosphate group, our second phosphate group. So now we're bound here, but now it's changed. If you remember, this second phosphate we add changes the head group into a pretty head group, so it's able to bind easier with things. Okay, now it's an ugly head group, but it is already bound here. Okay, so ATP, we try to add it in, it has a hard time binding to this ugly head group. Okay, it's going to take a lot of time for it to make that bond. So this is what's called the latch state. That's why I've kind of drawn it all wiggly, is because it takes such a long time. So this allows certain smooth muscles to stay contracted without using lots and lots of energy, right? Because we're not using up ATP. This ATP just takes a long time before it can make a bond. Okay, so once the ATP does make a bond, then we start over at square one, right? But until then, this latch state will stay. Okay, so an example of this would be a sphincter, right? You don't want to lose control of a sphincter, right? I know I've been able to hold my sphincters closed for a long time, and I've been grateful for that. I would be extremely embarrassed if I lost control of a sphincter at a time that was not a good time. So that's kind of an example of a smooth muscle that uses the latch state. Okay, with these tonic and phasic, our phasic sources of calcium, they come from a GPCR, a G protein coupled receptor. They cause the IP3 to come in, and that's what causes the calcium release. Okay. If you remember talking about IP3 and all that stuff in a past video. So that's where phasic gets its sources of calcium. Tonic gets its sources from extracellular channels. Okay. Now we're going to move on. We're going to talk about muscle sensory receptors. I hope you guys can follow along with me. I've kind of already got things drawn, but it's for the sake of time. So just think of this big outer red circle as our muscle fibers. Okay. Now they're known as extrafusal fibers. Now we have these little guys kind of inside here, near to the tendon, that are called intrafusal fibers. Okay, and they're known as muscle spindle apparatus. All right, and now they are stretch receptors. So whenever our muscles are being stretched, they are aware of that. They let us know kind of what's happening to our muscles. Okay, so as you can see here, I've got this big green thing coming in and innervating our extrafusal fibers, our skeletal muscles. Okay, these are the alpha motor neurons. Now we have these other guys over here, this little yellow one. coming in and innervating the muscle spindles, the intrafusal fibers. These are known as gamma motor neurons. Okay, so alpha, that means big, right? Gamma means small. But they are still motor neurons. The reason that these are important is when we come down here, we've got dynamic motor neurons and we have static gamma motor neurons. Okay, so this is kind of a blown up version of the spindle apparatus, the muscle spindles. Okay, so inside, near the middle, we have this big one called a bag. Trying to think how to word it. Bag, this is the chain fiber. Okay, now inside each of these, they have a sensory neuron. Now the bag fibers have 1A neurons, sensory neurons, and the chain fibers have 2A sensory neurons. Okay, so the 1A, it's responding to movement and stretch. 1A is a really big sensory neurons. It's the classification of a really big sensory neuron. 2A is a little bit smaller diameter. They're both myelinated, but it shows us or tells our body when our muscle is stretched, but it's now holding still. So if you take your arm and put it in an L position, and then you let it go straight, that whole time you're having 1A, 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 and then once you reach a flat arm, now it's saying 2A, 2A, 2A. Okay. Now there's some overlap in there. It's not exactly that way. Both are kind of firing, but you're receiving more information when you're moving from the 1A sensory neurons than the 2A. And then once you've kind of stopped moving, you're getting more information from the 2A. And that's letting your body know where your arm is in space. That's called proprioception. Okay. The reason that we need these gamma motor neurons is because, like I said, this is a stretch receptor. This muscle spindle is a stretch receptor. So what happens when you're contracting your muscles? You want these muscles to also contract, right? You don't want them to get loose and not tight, because you want to be able to know exactly when your muscle is starting to stretch. So what these gamma motor neurons do is they cause it to constrict or to contract when your other muscles contract, so that as soon as you start to stretch it, you have that automatic firing of these sensory neurons, letting your body know our muscle is stretching. This is really important in reflexes. Okay, so we're going to talk about reflexes now. We're going to talk a little bit about the Golgi tendon now, because in the muscle spindles, kind of an example, a really quick example is if you've ever gone to the doctor and they hit your patellar tendon with a little hammer looking thing. They're checking your knee jerk reflex. Okay, what that's doing is when they hit your patellar tendon with that hammer, it's sending the signal. Your muscle spindles are sending the signal that, hey, there's a stretch in my quad. And so what it's going to do is it's going to send these it's going to send the signal up saying, hey, we're stretching, right? And so it's going to say, hey, we need to contract. We need to go against this stretch. that we're having. So it synapses here, and then it causes our motor neuron to come and synapse in the quad, and so it's going to cause our quad to contract. That's why our leg kicks out a little bit. It's because those quads contract. With that small amount of pressure, the muscle spinders say, hey, we're stretching, I need you to contract, right? To kind of go against the stretch. So that's kind of what's happening. Now, this other pathway, in order for the stretch to, or the quads to be able to contract, the hamstrings have to relax, right? Because they're antagonistic to the quads contracting. So what happens here is there's an EPSP, and then you have this interneuron that causes an inhibitory response. So then it comes out and it goes to the hamstring, and it says, okay, hamstring, you need to relax. Both of these are alpha motor neurons, but one is doing an excitatory and one is doing inhibition. Okay. Pretty important to understand that. Now, the Golgi tendon reflex is kind of doing the opposite. The Golgi tendon reflex is really focused on your tendons, right? Our bodies are amazing. They can put up with a lot of force, and they're able to move a lot of weight. But because of this force that our bodies can generate, bones can be broken and tendons can be severed, right? And so our body has this Golgi tendon reflex that is in place to keep our bodies from doing too much and breaking something. So if you've ever been to a gym and you've seen guys lifting crazy amounts of weight, and all of a sudden they just drop their weight, they can't do anymore. That's their Golgi tendon reflex jumping in and saying, hey, like you're putting too much strain on, or too much force on this tendon, on this bone. And so it sets off an inhibitory to the muscles that are contracting, and tells it to stop, basically. Okay. So that's pretty much everything that we needed to cover today. Thank you all for watching, with going through this with me. I really appreciate it. We'll see you next time.